So the past few weeks we've been discussing different types of microbes, and we're going to round out our discussion on all these different types of microbes by including our last group, the viruses. So this will be all about viral structure, life cycle, and this is going to include information from chapter 5. So let's get a little backstory on viruses. Basically, viruses infect every type of cell on the planet, including bacteria, algae, fungi, protozoans, plants, and animals. So they're everywhere. Seawater alone can contain 100 million viruses per milliliter. So that's a lot of viruses chocked in a really small amount of seawater. For many years, the cause of viral infections was unknown. People didn't really understand what viruses were. Louis Pasteur postulated that a living thing smaller than bacteria was what was behind these viral infections. And he also proposed the term virus, which is Latin for poison. So people kind of had an idea it was something else, something that we hadn't really seen before. So another stride was made when Ivanovsky and Bergerac showed that a disease in tobacco was caused by a virus. So here we're seeing viruses infecting plants. And then Loeffler and Frosch discovered an animal virus causes foot and mouth disease in cattle. So at this point, we're seeing it in animals as well. And so they coined the term filterable virus. And that was what they found when they filtered these fluids from host organisms through these special porcelain filters that were designed to trap bacteria. But for some reason, the filtrate was still infectious, meaning whatever was in the fluids was passing through those filters designed to trap bacteria. So this was something that was smaller than a bacterium. And so hence the term filterable virus. And so this result proved that a cell-free fluid could contain agents that could cause infection, meaning these cells are smaller than any known cells and they could still cause an infection. So there's got to be something else other than what we currently know about. And they coined that term filterable virus to explain what that was. Okay, so we know they're smaller than bacteria. We know they infect a lot of different types of cells, but are they alive? That's kind of one of the questions that has persisted since the discovery of viruses. So there are two sides to this debate. Since viruses are not able to replicate independently from a host cell, they're considered to be not living things and should be called infectious molecules. And even though viruses don't exhibit most of the life processes of cells, they can direct them. So, it's not like they're dead. They can do things. They certainly can control a cell and get it to do what it wants to. So that's definitely not an inert or a lifeless molecule, but they are not able to do so without the help of the host cell. So it's better to describe viruses as active or inactive rather than alive or dead, because it's kind of a fine line since they aren't able to replicate independently, which is one of the characteristics of life. We consider them to be active or inactive for the purposes of this course. So we haven't really said what viruses are, other than the fact that they are not necessarily considered alive, that they're considered active or inactive, and we've said that they infect a lot of different types of organisms, and that's about it, right? So what are they? So this is a nice table that summarizes the key characteristics that set viruses apart from anything that we've discussed so far in this course. Basically, they're obligate intracellular parasites of different organisms. Obligate meaning they have to be in that particular relationship. Intracellular meaning they're inside of the host cell. Parasites meaning this is going to be a relationship that's beneficial for viruses, harmful for the host cell. They're ubiquitous in nature. They're everywhere. They're all over the place. They're ultramicroscopic, meaning they're very tiny. They are not considered to be cells. These are acellular in nature. They don't independently fulfill the characteristics of life. As I mentioned before, they're not able to live on their own. They depend solely on the host cell. Outside of the host cell, they're just inactive macromolecules kind of bouncing around. Then once they get inside the host cell is when they can propagate themselves. The basic structure is just a protein shell surrounding a nucleic acid core, so it's a very simple structure. That nucleic acid can be DNA or RNA, but never both. Now, this is the first time that we are seeing an organism that can have RNA as its genetic material, so you can see viruses are very unusual in this fact. The nucleic acid can take a variety of forms. It can be double-stranded DNA, it can be single-stranded DNA, single-stranded RNA, or double-stranded RNA. So this is a property very unique to viruses. The molecules on the virus surface impart a high specificity for attachment to host cell. What does that mean? That means that there are molecules on the surface of viruses that are honing in on particular host cell receptors, meaning that they're specific for these host cell receptors and they can only dock at these host cell receptors. So they're going to be specific for certain types of cells within the host. These guys basically aren't bringing anything to the party. They lack the enzymes for most metabolic processes. They lack machinery for synthesizing proteins, and they basically multiply themselves by taking control over the host cell and regulating the synthesis and assembly of new viruses. So they're going to take over the ribosomes. They're going to take over the DNA replication materials, those types of things. So viruses are very simple, but they're also very complicated in the fact that they have the smarts to do all of this within the host cell. So I mentioned on the previous slide that viruses are ultramicroscopic, but what does that mean? How small are they really? So they are the smallest infectious agents that are known out there. So they're very small. But some of the largest viruses are about as large as some of the smallest bacteria. So I have this figure in here so you can visualize that. The big pink rectangle and the big purple circle, those are some different sized bacteria. So those are some bigger bacteria. Then where you have the overlapping of the two black circles, that is those two little pink rectangulars. Those are going to be a small type of bacterium. Those are actually rickettsia. So these are a small bacterium. The virus that's next to it, mimivirus, that's actually a large virus. So some of the largest viruses are about the same size as small bacteria. So don't think that all viruses are these ultra tiny things compared to bacteria. It really depends on the virus. Most are very small. Some are very, very small, and some are larger. In fact, they're about the size of some small bacteria. Okay, so just to keep that in mind, there's a range. So let's talk more about viral structure. I mentioned a few slides back that viruses are not cells. They are acellular, and basically they're just a protein surrounding their nucleic acid. So viruses aren't going to be like anything we've seen so far. They're not prokaryotes, they're not eukaryotes, they're their whole own thing. A fully formed virus that's able to establish an infection in a host is known as a virion. So this is an important term for you to know. And a virion is made up of two major parts. We have the protein part that's covering the nucleic acid, and that's known as the capsid. Then we have the nucleic acid molecules that's going to be in the center and the core of the virus, and those can be DNA or RNA, as I mentioned earlier. So basically, it's going to be a really simple model for virus structure. You're going to have this central nucleic acid surrounded by this protective protein coat called the capsid. So let's talk a little more about the outside covering, and let's start with the capsid. So the capsid is going to be that protein that is protecting the nucleic acid. It is composed of these identical subunits called capsomeres. And we have two major types based on the shape of the capsid, and those two types are helical and icosahedral. Now, an envelope is going to be an optional outer layer that covers the capsid. This isn't going to be seen in all viruses, just some viruses. The envelope is usually made from a modified piece of the host cell membrane, and it obtains that piece of the host cell membrane when it's leaving the host cell, and we'll see that in a few slides. The regular host cell membrane proteins are replaced with viral proteins, and so everything just flows together really nicely for the virus. Spikes are going to be projections on the capsid or the envelope that allow viruses to dock with the host cells. Remember a few slides back I mentioned that viruses have specificity, meaning they can't just go into any host cell. And part of this is the interaction between the projections on the capsid or the envelope, those spikes, or some other type of projections, and the host cell receptors. So that interaction is what helps get them into the host cell. Okay, how about what's in the middle then? What is at the core? Remember, it's that genetic material. So viruses can contain DNA or RNA, but not both. They're not going to have both at the same time. The number of viral genes is quite small compared with that of a prokaryotic cell or a eukaryotic cell. They only are going to be carrying around the genes that they need to invade the host cell and to kind of take over the host cell machinery. So they're going to travel pretty light in terms of their genetic material. Now, I mentioned earlier that viruses have like a lot of different types of configurations of DNA or RNA. You can see single-stranded DNA, double-stranded DNA. It can be linear, it can be circular. In RNA viruses, you're going to see double-stranded, but most often you're going to see single-stranded RNA viruses. You've got a positive sense strand of RNA, which means basically that the RNA is the mRNA that we would use in transcription translation, and that means it's ready for immediate translation. The negative sense RNA means that it must be converted to the complementary form before translation can occur. So that's something that's a little different, something we haven't seen so far. And then we also have RNA viruses, which are known as retroviruses. They actually bring a little something extra with them. They carry their own enzymes to create DNA out of their existing RNA. So that's something that's going to be quite different than we've seen so far. It's important that you have a frame of reference for these different types of viral nucleic acids. So we've talked about there's double-stranded, single-stranded, all these types of things. So this is just a nice table that summarizes a few examples of these different types of genomes that a virus can have. And so, for example, the variola virus and the herpes simplex 2 virus, those two viruses are examples of double-stranded DNA viruses. Parvovirus, that's an example of a single-stranded DNA virus. Poliovirus, that's going to be a positive sense single-stranded RNA virus. Influenza is going to be a negative sense single-stranded RNA. Rotavirus, which is really common in cases of gastroenteritis, which is like a GI distress. We'll get to that later this semester. That is going to be a double-stranded RNA virus. And then HIV is one of those retroviruses that I mentioned. It's a single-stranded RNA virus that, using reverse transcriptase, the enzyme it brings in with it, can be changed into double-stranded DNA, and then it incorporates into the host cell genome. Now, this one's important because HIV, we know, has a lot of healthcare implications, and we'll be discussing that later this semester. Okay, well, that seems pretty simple so far. There's the protein capsid, there's the nucleic acid inside, some of them might have envelopes. Is there anything else that we're missing? Well, some viruses do carry a little extra with them. They carry some enzymes in with their central core to help them with specific viral operations. These are going to be enzymes that they are not going to be able to find outside of their own being. So it's not going to be something a host cell would have. So some viruses carry some polymerases, which help make more genetic material. Some carry replicases to help copy RNA. And some use this reverse transcriptase that I mentioned earlier, like HIV, and that's to synthesize DNA from RNA. And then some take souvenirs from the host cell to use for their own self later on. So retrovirus can sometimes borrow the host tRNA molecules, which are useful in translation. So that's just something to keep in mind, that the general simple structure is just a protein capsid and the nucleic acid. But some viruses do carry a little bit of extra with them depending on the virus. So how do these simple acellular viruses go about making more of themselves? Basically, there are 5 main phases in the life cycle of an animal virus. So we're going to focus on an animal virus. That would be a virus that infects animal cells. So we're going to look at those particular types of viruses. And there are these 5 main phases: adsorption, penetration and encoding, synthesis, assembly, and release. Now, the time it takes for these 5 phases to happen varies depending on the virus, and it can be anywhere from 8 hours to 36 hours, again, depending on the virus. So table 5.4 is a nice visual accompanied by some great written descriptions of how this general life cycle works. So let's break each of those steps down into more detail. So step 1 is going to be adsorption. Now, a virus can invade its host cell only if it can make an exact fit with the specific host cell molecule. It's not going to go ahead and just go match up with any host cell. It's got to have this nice fit. And so if you see in the figures there, we have an enveloped virus that has these spikes all over it. We've got a non-enveloped virus that has these capsid spikes on it, and they're binding with these receptors that are on the host cell. You can see they have a specific shape, and the viruses are fitting into there. Both enveloped and non-enveloped viruses must find receptors to match up with in order to successfully attach to the cell or adsorb. That's another way we can say attach. Now, the host range is the limited range of cells that a virus can infect. Depending on the virus is going to depend on what type of cells it can infect. For example, hepatitis B virus, it's going to infect the liver cells of humans. The hepatitis B virus has receptors on its surface that only match up with the receptor cells of hepatocytes or liver cells. Cells that lack compatible virus receptors are resistant to absorption and invasion by that virus. So that's good news for the host, right? If you don't have that particular host cell receptor, you're not going to have that virus dock with you, and you won't have the life cycle progress. So adsorption is a really important first step. Now, tropism is the term that we use to describe the specificities of viruses for certain tissues based on this whole matching up with the host cell receptor business. Okay, you are the virus, and you have completed step 1. You have successfully attached or adsorbed to the host cell. So now what's the next step? Getting in, right? Remember, you are an intracellular parasite. You need to get inside the host cell. So the way you do that is to penetrate the host cell and get in. So there's 2 ways to enter the cell. There's endocytosis, which is on the left-hand side of the figure, and there's fusion, which is on the right-hand side. Endocytosis is a way where the virus is engulfed by the host cell and brought in through a vesicle or a little bubble. Fusion is for enveloped viruses only. Basically, the envelope just fuses with the host cell membrane and frees the nucleocapsid into the cell. So endocytosis is bringing the virus in through a bubble. Fusion is enveloped viruses only. The envelope just kind of meshes with the host cell membrane, and then it releases the nucleocapsid into the cell. Now, once inside, all of the viruses need to get comfortable and take their coat off. Remember, the coat is the capsid, and we need to take the coat off because we need to expose those nucleic acids, because that's where the important information is stored. That information is going to help the virus replicate itself. So uncoating is the process of the virion shedding its capsid to liberate the nucleic acid and let it get to work. Okay, you've broken into the host cell's layer, and now you want to replicate yourself, right? You want to get it kicked off to where you start making more virions. So that is step 3, synthesis. And this is the phase where the virus's genome, either DNA or RNA, is replicated and proteins for the capsid are made. And that's really all there is to the virus, right? Just the protein capsid and the genome. So synthesis is where those things are going to be made. Now, for DNA viruses, they're going to enter the host cell's nucleus and replicate themselves there. For RNA viruses, this process is going to occur in the cytoplasm. Why in the nucleus versus the cytoplasm? Well, for DNA viruses, the DNA replication machinery is in the nucleus. So you've got to go where the machinery is available. For RNA viruses, that is in the cytoplasm. Now, retroviruses turn their RNA genomes into DNA. We've mentioned that a couple of times, so that's important to know that that's a really important step in the retroviral life cycle, is turning their genome from RNA into DNA. So once you have everything made, you need to put it together, right? So step 4 of the viral life cycle is going to be assembly. And this step actually puts together the new viruses using the parts that have been manufactured in step 3, and that synthesis process. And so all proteins for the capsid are made in the cytoplasm, regardless of if you're an RNA or a DNA virus. And so you've got to get those parts joined up with the nucleic acids. So for DNA viruses, the proteins migrate into the nucleus to assemble with nucleic acids. For RNA viruses, the proteins and nucleic acids join up in the cytoplasm. So now you're all put together, you're still a virion, you're all put together, and what do you want to do? You want to break out of the host cell, right? You've done what you came to do, it's time to move on to a new host cell. So step 5 is going to be released. And the number of viruses released by infected cells is variable, and it's basically controlled by the size of the virus and the health of the host cell. And there's only 2 ways to get out. There's lysis and there's budding. Now, lysis is usually what's going to happen if you are a non-enveloped virus, meaning you don't have an envelope. It is the bursting of the host cell that usually results in the host cell dying. Budding is going to be the release of virions through the host cell membrane, and this is the stage that viruses get their envelopes, and only enveloped viruses are released that way. So, budding is the only option for enveloped viruses. They're not going to get out through lysis. They have to get out through budding. So based on what we've discussed so far, I'm sure you're getting the picture that a viral infection of a cell is not necessarily good for the host cell. So any kind of viral damage that is done to the host cell is known as a cytopathic effect. And so these are changes in the host cell that are made by the virus during the course of a viral infection. Now these cytopathic effects can be very minor or they can be fatal for the host cell. Some examples include changes in the shape or the size of the host cell, formation of inclusion bodies. Basically, an inclusion body is just going to be this compacted mass of viruses or damaged cellular organelles from the host cell. And you can see that in the photo below, those dark bodies indicated by those lines that say inclusion bodies, those are going to be an example of an inclusion body. We can also see the formation of syncytia, which are large multinucleated cells that form from multiple host cells fusing together. And so that's a common cytopathic effect of a viral infection. Okay, so cytopathic effects are effects on a particular host cell. But what about the whole entire host? Let's look at the types of viral infections that can be present. So you can have an acute infection that's just going to run its course very quickly, but you can also have some other types of viral infections. You can have a persistent infection where basically the cell harbors the virus but isn't lysed right away. So basically the virus is hiding out in the cytoplasm or it's incorporated into the host cell DNA as a provirus. So that's what we call the form of the virus when it's incorporated into the host cell DNA. Persistent infections can last from a few weeks to a lifetime, and it can take a while for symptoms to show. They can show up weeks later or they can show up years later. But detectable viruses are found. They might be in very low levels, but you are able to detect viruses in persistent infections. And examples of a persistent infection would be an HIV infection or an HPV infection. A latent infection is a little bit different. Basically, viruses go into a period of inactivity and can also be incorporated as a provirus, as we mentioned with persistent infections. But then they emerge later due to a stimuli such as stress or poor health. During that time of inactivity, we do not see symptoms and we do not see detectable viruses. They are truly hiding out. They can cause recurrent disease. So basically, you have like a flare-up. You have symptoms followed by no symptoms followed by symptoms again later on. And so an example of this would be something like cold sores or genital herpes, something that will go away but comes back and then goes away and comes back. Or something like herpes zoster virus, where you have chickenpox as a child and then it hides out and then you can get shingles later on as an adult. Viruses can also cause oncogenic effects. This is when a viral infection causes mutations in the host cell DNA that results in cancer. So oncogenic viruses are ones associated with cancers. Examples of an oncovirus are papillomavirus, HPV, and hepatitis B virus. So earlier I mentioned that viruses can infect all type of organisms, and bacteriophages are going to be viruses that infect bacterial cells. And this is important because bacteriophages can confer pathogenic properties to the bacteria that they infect. They have similar life cycles to the animal viruses, but it's actually a little bit simpler. The nucleic acid is injected, so there's no encoding involved. And there are two types of bacteriophage life cycles. We have the lytic cycle and the lysogenic cycle. Now, the lytic cycle is extremely similar to the animal virus life cycle, and the release of the virus occurs through lysis, bursting of the host cell, which then kills the host cell. The lysogenic cycle is a little bit different. What happens in the lysogenic cycle is that the viruses enter the bacterium, they insert into the bacterial chromosome, forming a prophage. Sometimes we have induction occurring, which is where the prophage activates and begins replication, and then the lytic cycle occurs. And this is less damaging to the bacterial host cell than the lytic cycle is. Lysogenic viruses are important because they produce a phenomenon called lysogenic conversion. And basically those prophages that are incorporated into the bacterial chromosome can pass along traits that let the bacteria produce a virulence factor, a toxin or an enzyme, something that's going to help them with progression of disease. A really great example of lysogenic conversion occurs in the pathogen Corynebacterium diphtheriae, which causes the disease diphtheria. Now, the seriousness of this disease is determined by the presence of the diphtheria toxin, and the diphtheria toxin is something that is passed to the bacteria through this lysogenic bacteriophage. So since we've been discussing viruses, which are non-cellular or acellular infectious agents, let's also discuss some other kind of weirdo infectious agents out there, the prions, and then also virus-like agents. Now, prions are made entirely of proteins. They contain no nucleic acid, so they are super weird as far as infectious agents go. How they replicate themselves is under study. There seems to be some theories out there, but we're really not sure how they replicate themselves since they don't have any nucleic acid. They are associated with causing diseases known as spongiform encephalopathies, which basically cause your brain to look like sponge, looks like it has holes in it. And these diseases are progressive, fatal, and always affect the brain, and most human versions are somehow genetically linked. And so one very well-known prion disease in humans is Creutzfeldt-Jakob disease, or CJD. It's very rare, but it is caused by prions. Variant Creutzfeldt-Jakob disease, or VCJD, is a human spongiform encephalopathy that is linked to bovine spongiform encephalopathy, or BSE, and that's found in cattle, and you might know it as from its common name, mad cow disease. And so there appears to be some type of link between the genetic variation and the bovine form for the variant Creutzfeldt-Jakob disease. There are other non-cellular infectious agents known as the virus-like agents, and we call them the satellite viruses. They're actually dependent on other viruses for replication. So we have the adeno-associated virus, or AAV, originally thought that it could only replicate in cells infected with the adenovirus. It can also infect cells that are infected with other viruses or have had DNA disruptive for some reason. So once was only associated with adenovirus infected cells, but now we know that this particular virus just needs some other virus in the vicinity in order to replicate. We also have the delta agent. It's just a naked circle of RNA, no envelope, no protein capsid, nothing like that. It's expressed only in the presence of the hepatitis B virus, and it can actually worsen the liver damage effects of hepatitis B. Again, these are satellite viruses. So viral infections greatly impact human health. They cause billions of infections worldwide each year. They cause acute infections. I mentioned those earlier. Those are going to be just ones that occur quickly and resolve quickly. So they run their course quite quickly. And those are things like colds, chickenpox, influenza, warts. Geographically prominent infections are things like dengue fever, Rift Valley fever, yellow fever. These are infections that are only happening in certain parts of the globe. Viral infections with high mortality rates are things like HIV and rabies. Viral infections that cause long-term debility are things like polio and neonatal rubella. And then viruses possibly connected to chronic afflictions, which is a very hot area of research right now. So they think that viruses are connected with these chronic afflictions, things like diabetes, multiple sclerosis, Alzheimer's disease, obesity, and specific cancers. So this is something that people are researching because until we can completely rule out that they are virally caused, then we can't be quite sure. So that's a hot area of research right now. So how in the world do we treat viral infections with all these billions of infections occurring worldwide each year? Well, antibiotics don't work on viruses. So things that work on bacterial cells aren't going to work on viruses. We have antiviral drugs that generally block viral replication, but they can have severe side effects because remember, the viruses are in your cells, in your host cells, so that can have problems for you. We have natural protection against viral infections within our immune system, known as interferon. We also have synthetic versions available for the same thing. We have synthetic interferons available for some protection of viral infections. And of course, vaccines vaccines are key in protecting us from specific viral diseases. Of course, not all of them, but from a lot of them. So that's how we kind of manage viruses in human health. So let's focus on a viral disease. Let's focus on shingles. Shingles is a latent disease caused by the varicella-zoster virus. This same virus causes the acute disease chickenpox. So after an individual recovers from chickenpox, the virus goes into a latent form and basically hides in the nerve cells. So basically it can't be detected. As you age, as your immune system changes, as you are subjected to stress or other unknown factors that we can't quite pinpoint now, the virus becomes reactivated, viral replication occur, and symptoms begin to show. Now, symptoms include tingling, burning, pain, preceding a blister-like rash, which then crusts over. Now, the pain associated with shingles can be very severe, and that's because the virus affects the nerve cells. And so, since your nerves are being affected, there's more pain associated with this than there is with chickenpox. Since the nerves are affected, you can have nerve damage, which is referred to as post-herpetic neuralgia, and that can cause chronic pain for the duration of your lifetime. To treat this disease, we use antivirals like acyclovir, famciclovir, and valacyclovir. It can shorten the course of the disease, but it has to be started really early, within 72 hours of symptoms, in order for it to be effective. And the antiviral is going to shut down the replication of this particular virus. There is a vaccine that's recommended for adults that are more than 65 years old, which is the most susceptible population for this disease. It can prevent shingles, and it reduces the occurrence of that post-herpetic neuralgia, which can be the cause of chronic pain in someone who's gone through shingles. So that will sum it up for viruses. Be sure to bring any questions that you might have to class. See you next time.